different from anything that had been seen before to be confident about them. And that's in the human gut, which is probably the best studied microbial community. If you start moving out to more and more difficult environments with more and more diverse and diverged microbial biochemistries, there's a lot more yellow here. So there's a lot more genes that we can't easily tell what they are when we first see them, let alone what they're doing. So again, there's a lot of glass half full, glass half empty here. A lot of those yellow genes, if you spend just a little bit of time, we can give them some pretty good annotations. But there is a long tail at many of them. These are genes that will keep us all employed for a long time, trying to figure out what the more and more idiosyncratic biochemistries are doing. So part of the work that we've done to help capture the low-hanging fruit is as part of the Human Microbiome Bioactors Resource, or HMBR. This was a collaboration with the group shown here, Romnick and Wendy here in Boston, Owen and Anoop at the University of Maryland. And the goal of the HMBR was to characterize novel bioactivity defined in a similar way as biochemical function that influences or responds to human health. This was specifically in the gut and specifically for inflammation in this case. This was funded by the NIH here to look at gastrointestinal inflammation. And we focused on three different kinds of bioactivity, either phenotypes that were caused or influenced by whole microbes being present or absent, direct mechanisms of biochemistry where microbes would modify or create a small molecule, so biochemical bioactivity, and then indirect mechanisms, including things like microbial protein production. Do you make a protein that then influences, say, the immune system to respond, or so on and so on? So focus on just a subset of these today, particularly looking at protein bioactivity in this case. So one of the first things we wanted to do when we looked at this big pool of uncharacterized proteins that we find even in the well-studied human gut microbiome is figure out, well, which out of these hundreds of thousands of proteins, of uncharacterized proteins, are most important by some definition? Which ones do we think we should study in more depth? So a postdoc at the time, Yen-Song Zhang, put together a workflow specifically to prioritize potentially bioactive microbial gene products. And I won't go into the computational details too much. The system named Metavibula for this complicated acronym consumes metagenomes, assembles them into contigs, and finds open reading frames, genes, within those contigs. They're clustered by nucleotide identity and then protein identity and sorted out into protein families that we've seen before and we pretty much know what they do, all the way down to really novel protein families that, again, don't look like anything we've seen before. Metavibula combines three criteria to then prioritize which of these proteins are potentially bioactive. One, of course, is based on sequence information, homology, domain annotations, anything that you could traditionally find from a sequence. Some of those prioritization, though, relies on an environmental or phenotypic properties. Do these proteins co-occur with a health phenotype or an environmental phenotype or exposure of interest? And then the third set of criteria come from ecology. How are these protein families distributed? Are they prevalent? Are they abundant? Do they appear in everyone or just certain individuals? So combining these three types of criteria, we can try to guess which proteins out of many uncharacterized ones might be more important. So when you look at this in, again, the human gut microbiome, and I'll use in this case about 1,600 metagenomes, shotgun metagenomes, from the second phase of the Human Microbiome Project. So these are drawn longitudinally from around 130 participants with stool collected every couple of weeks over a year. And again, I won't get into the phenotypes too much, but in the HMP2, we were looking at the inflammatory bowel diseases, Crohn's disease, and ulcerative colitis as compared with non-IBD controls. So you can keep this in mind as sort of a high-level overview of the data set. We'll come back to this same data a little bit later. 1,600 shotgun metagenomes longitudinally from 130-ish individuals, half Crohn's disease, a quarter ulcerative colitis, and a quarter non-IBD controls. When we look at all of the complete open reading frames from all of the assemblies from all of these metagenomes, we end up with about 45 million individual genes. Now, these pretty quickly cluster down to only about 2 million nucleotide sequence families. So these are genes whose nucleotide sequences are very similar, and roughly 1.6 million protein families. So when we then translate those nucleotides to amino acids and continue to cluster them. Of these 1.6 million protein families, only about 500,000 of them are characterized by the definition that I raised earlier. Another 400,000 of them have strong homology to something that has been seen before, but which again has not actually been characterized. 500,000 or so of the gene families have some homology to something that has been seen, but it's a modest amount of homology and not enough to be confident about what function that protein is carrying out. And again, even in this very well-studied population, there's still several 100,000 proteins that are yellow, that don't have any significant homology to anything that's been cataloged before. And this is after quality control and looking for just confidently annotated gene families, so we're pretty sure these are real. So we prioritized these using MetaVibula, and again, I won't go into a lot of detail, but we tested a couple of the top predictions, some of which, for example, were novel pillin families that were implicated in IBD-associated inflammation. And it turned out nature had done a nice genetic experiment for us. Different strains of E. coli already carry different combinations of these pillin families and did prove to be differentially immunogenic depending on which pillin families they had. Similarly, another whole family of proteins that were prioritized were homologs of human von Willebrand factors, which are proteins involved in blood clotting in eukaryotes. So we looked at those and thought, okay, it's got to have something to do with adhesion. That would make sense. And we tried to work with genetics in a Bacteroides model in this case. And finally found a biofilm phenotype in the gene family knockout, specifically in the presence of mucin. It drove us crazy trying to find a phenotype for a while. So these indeed were proteins that are bioactive in the protective formation of Bacteroides biofilms in the gut, specifically at the mucous membrane. So I've gone through this pretty quickly since this work was published a little while ago, and we've just recently finished some follow-up that helps to specifically characterize some of these prioritized proteins, because just because we think a protein might be important doesn't necessarily mean that we know why. So there are different computational methods that have been used for many, many years now to predict or annotate protein function, biochemistry, regulation, structural proteins, function by the definition I showed earlier, mostly in isolate organisms. So for something like Saccharomyces cerevisiae or E. coli or mice or human proteins, computational methods have been designed to integrate different types of evidence from high throughput experiments, from genomics, from the literature, from structural information, and use those different evidence types to predict protein function. So Yansong wanted to apply this idea to whole microbial communities, and again put together a complicatedly named system, in this case called Fugasum, to predict function for uncharacterized gene families, specifically in microbial communities. And here I will spend a little longer talking about the bioinformatics, since Fugasum relies pretty heavily on meta-transcriptomics in addition to metagenomics. The idea being that you can get more functional information out of functional activity in the form of differential regulation or expression, along with sequence-based information that you can get from metagenomics. So Fugasum will consume a collection of both metagenomes and paired metatranscriptomes, again cluster those into protein families just like we saw before. But now what it does is separate them out per taxon. So it'll collect a bag of protein families, a pangenome, associated with each taxon in a collection of microbial communities, and build interaction networks for each data type. So an interaction network for protein domains might literally look like protein-protein interactions within each microbe. But something like, say, homology turns into a network where the edges represent sequence similarity. Something like an adjacency analysis turns into a network where if two proteins are close to each other on a contig in an underlying genome, then they have a connection between them. And maybe most importantly over here, a network built on gene expression data on metatranscriptomics is built out of co-expression. If two proteins co-vary together, if they're turned on and turned off at the same time, then they probably have something to do with each other functionally. Fugassem then uses two layers of machine learning to combine all of these networks, again, once per taxon. The first layer essentially normalizes each data type. It'll say, if two proteins are co-expressed at a level of 0.6, I trust it this much. If they're co-expressed at a level of 0.3, I trust it this much, and so on and so on. And do the same kind of normalization for different measurements within each data type. The second layer takes those normalized networks per data type and combines them. It'll essentially learn how much to trust each whole data type. So if you see a high measurement in metatranscriptomes, maybe that means more or less in terms of functionality than a high measurement in terms of sequence similarity. And what this then outputs is a network that is turned into a set of protein function predictions for each of the uncharacterized proteins we see in each taxon's pangenome. So again, this is exactly the same as what's been done for single organisms for a long time, and thus we're able to compare Fugassem's behavior on whole microbial communities and its accuracy there to either methods that were developed to do kind of the same thing for single microbes and to databases that also integrate many different data types for single microbial genomes. So what you're looking at here is a performance measure using cross-validation across the set of microbes with HMP2 metatranscriptomics and enough isolate-based gene ontology annotations that we can evaluate them, that we know a ground truth, so we can hold some of them out and measure whether we predict them back correctly or these other systems predict them back correctly. So you can see, first of all, that Fugassem is able to generate predictions for many more organisms than other methods because we can essentially work with anything we see in a whole microbial community. So the rows have more colors in them. If you look at the top here, metatranscriptomes are sort of moderately informative. If we only use metatranscriptomes by themselves, we still get a lot of predictions right. Homology starts to do even better because we are now able to leverage homology among many, many different versions of a protein family throughout a community, rather than relying on just the subset of versions of a protein family captured by isolates up here, for example. And something that really surprised us at first is this bottom row here, where we were getting just really good predictions by integrating data types. And we sort of puzzled over why that was for a while, and it turns out that the combination is nonlinear. If you co-express together, yeah, you probably function similarly. If your two proteins are kind of homologous, then yeah, they might do something related to each other. But if both of those things are true, then you can actually be really confident that, yeah, those proteins are functionally related. So the different data types are quite synergistic. Another thing that really surprised us when we look at how much new stuff we predict for the pangenomes of different organisms, of course, we can get predictions again for organisms that don't even have isolates. We can make some predictions. We can't necessarily know ahead of time whether they're right or not, but we can make predictions for anything that we can pull data out of the microbial community. But some of the greatest number of new predictions were coming up for these really well-characterized organisms like E. coli. And same thing, we looked at that and thought, no, that's wrong. That can't be right. Turns out that even E. coli, which is so well studied, has such a large pangenome in the wild that we'll see tens of thousands of additional E. coli gene families in the human gut that, of course, are not captured by K12, the laboratory strain. And since those wild E. coli are pretty similar genetically to characterized E. coli or closely related organisms, we actually have quite a bit of power to make confident predictions for them. So this is what I mean by low-hanging fruit, where even for well-studied organisms in a well-studied environment, there's a lot that hasn't been characterized, but which we can pretty confidently analyze given a little bit of computing. And then just to show that there are actual individual genes underlying all of these high-level evaluations, we can zoom all the way in to see individual genes in specific network neighborhoods for specific pathways. So Faecalibacterium prausnitzii is sort of a good bug. It's a gut microbe that is often associated with being anti-inflammatory, associated with health. And in F. prausnitzii, each of these orange proteins is an uncharacterized protein that has some homology, but which we don't know what it does. Each red dot is a specific protein that has no significant homology to anything previously studied. And here, even for systems like phage defense or the CRISPR system in FPROUS, we can start adding in genes that are clearly part of these protein complexes or subpathways within CRISPR defense in this particular case. Same thing over in more pathobiont-like organisms. So Hungatella is a bug that's fairly recently been associated with colorectal cancer. And here, both some of its motility and structural proteins that are likely involved in pathogenic behavior, and B vitamin biosynthesis and utilization, which is kind of a privileged micronutrient in a lot of human-associated communities. Here, Hungatella is not a particularly well-studied organism, and we can start placing more and more of its protein families into potential pathways to help understand how it behaves. So I'll switch gears there. We've been working a lot to continue following up different types of bioactivity in the HMBR. I've not talked very much today about some of the biochemical characterization we've done, but I want to take the last few minutes to talk about a different kind of uncharacterized or undercharacterized protein function that we're working on as part of the Human Virome Program, or HVP. This is a relatively new program. The Common Fund at the NIH launched it at the beginning of this year. And like we mentioned at the beginning, I'm delighted to coordinate or to help run the coordinating center there with Owen White and his team at the University of Maryland again. So we're responsible for making sure to gather and share all of the data and resources generated by the consortium, which consists of a combination of 5 big virome characterization centers who are tasked with making data that describe the body-wide human virome, specifically in health. So what viruses are normally associated with human-associated microbial communities in the absence of disease. And there are 10 different studies, 5 of which are tasked with characterizing specific biological functions of viral communities, the other 5 of which are methods-oriented, either computationally or experimentally. So if the VCCs are making lots of data and the smaller projects are either helping to interpret those data either computationally or biologically, our goal at the coordinating center is to really help get all of these resources out there. As part of the HVP, something that I'm excited about computationally is being able to better detect viruses in microbial communities in the first place. They're, of course, very difficult to isolate in culture, much more so than bacteria. They're great. Let's see, am I back now? There we go. They're great virus-like particle enrichment protocols, so VLP enrichments, that will physically take a sample and try to sequence just the viruses. These can be very difficult to execute. Sometimes they work great, sometimes they're more difficult depending on the sample, and they can enrich or deplete different viruses, so you no longer get a sort of even quantification of your community. On the other hand, they've been great for viral discovery. So we suddenly have hundreds of thousands of metagenome assembled viral genomes, or VMAGs, from several of these great publications and accompanying databases that we can leverage now to help find viruses more easily in other types of communities. Because most of the time, most reads in most communities don't assemble, whether that's for viruses or otherwise. So we've been trying to put these pieces together to better detect and quantify viruses, even in normal shotgun metagenomes and metatranscriptomes, since we can look for RNA viruses as well. So the last funny name I'll talk about today is baklava, which again stands for a complicated acronym. What it does is profile viral taxa out of metagenomes or metatranscriptomes, whether or not they've been VLP enriched. It uses some technology from our metaflan profiler to do this by constructing viral nucleotide markers. It uses some technology from our human functional profiling software. But instead of looking for protein function in this case, we're using proteins to find more diverged viral sequences. Since viruses evolve very quickly, just nucleotide matches are sometimes not enough to recognize them. And then finally, we have a third arm of baklava that's based on a deep learning system that will work with assemblies and directly classify contigs, which is what a lot of the current viral isolate or MAG detectors do. So we combine these three different approaches to generate an integrated viral community profile for each input metagenome or metatranscriptome. I think, again, I'll skip over some of the computing, looking at the amount of time left. The short version, the secret sauce is that we'll search each genome for sequences that are unique to that genome after quality control and clustering, save all of the sequences that are unique, and thus for viral families that have enough unique sequences, now every time we see one of those sequences in a new community, we can be confident that we have seen that virus. And this results in very high recall and precision. So much higher, similar precision to a lot of the assembly-based methods and much higher recall than assembly-based methods. So we have near perfect recall and precision for known viruses, pretty good recall and precision even for novel viruses, and a good low false positive rate for non-viral sequences. And here, these are two different sets of synthetic communities. So this is showing for different distributions of microbes in a synthetic community, we still do a pretty good job at finding the viruses accurately. And to wrap up with this one last example, also from the HMP2 inflammatory bowel disease metagenomes, here we're using the same 1,600 shotgun metagenomes and bringing in another 800 or so shotgun metatranscriptomes to now find and analyze the viruses there. So one of the oldest observations in the IBD microbiome is that during disease, especially during disease flares, since IBD is a remitting disease, recurring remitting disease, where patients will go through flares of activity, during those flares, the gut microbiome loses diversity. And we see the same thing in the viral community, but it's true even outside of flares. And that's not true for the rest of the microbiome. So the bacterial portion of the gut microbiome is pretty stable outside of disease activity in IBD. The virome still responds and shows reduced diversity. And that's true within the overall disease population. And it's true when we compare compare across disease populations. So there's more overall variability among IBD patients' viromes as well. Interestingly, that variability is extremely personalized, even more so than the bacterial portion of the microbiome. Some individuals carry viruses that are quite common and seen in pretty much everyone. Other individuals carry almost entirely unique viruses. What you're looking at here is 1 bar per individual, and the bars represent the total viral community. Darker colors are the portion of viruses that are shared among more people, and lighter colors are those that are not. So these individuals carry pretty much only their own unique viruses. Other individuals only carry viruses that are pretty common across the population. And again, I don't think I'll quite have time to go into it, but we can drill down to the level of individual taxa, either examples here from bacteria, or examples here from the viral profiles that are individually associated with disease activity or disease diagnosis. So if you just look at this slice off the top of the most IBD-associated viruses, they're basically all phage. Most of what we see in all of our human samples are phage. Many of them here are crassphage, which are typically Bacteroidetes phage. A couple of them, like Foxway virus, is an E. coli phage, Totatus virus, and many of them are unclassified. And most of them are specifically lost in IBD. You can see that they're absent during CD and UC, which again mirrors what we see in the bacterial communities. And I didn't show anything even quickly from the RNA viruses, so stay tuned. We're just getting those analyzed. Like I said, I'll wrap up with a quick blurb about our Microbiome Center here at the Harvard D.H. Chan School of Public Health. Too many words. The HCMPH, which is easier to say, is our umbrella community for different types of microbiome science, whether that's epidemiology, nutrition, computing, basic biology, and immunology. I co-direct the center with Wendy Garrett, who focuses on mouse models of the microbiome and their molecular and immunological responses. Among many things that we do is offer both sample collection services and bioinformatic analysis services specifically for microbial community studies. And that's true whether it's the human microbiome or not. So if you have a need to collect tens of thousands of stool samples or analyze tens of thousands or even dozens or tens of microbial communities, you can get in touch with either the collection core or the analysis core. We have both kind of direct kit construction services and consulting services. And save the date if you're interested. We also have both an annual symposium coming up next May. And this past year, we started running a local workshop for a week-long, more intensive tutorial on computational methods for microbial community studies. So we're pretty sure about the two dates. They're not set in stone yet, but if you want to keep an eye on these for next year, we'll post more information as soon as we have the dates locked in for both of those. I know I'm a little bit over time, so I apologize. I've skipped over calling out some of the other faces that you saw on the slides who are either our fantastic collaborators over here on the right or folks from the lab who actually did all of this work. So if there's still time, I'm happy to take any questions. Thanks again so much for the invitation and setting all of this up.
Thank you. Many thanks, professors. Thank you. We are grateful for this new insightful topic, especially the protein bioactivity and the new project, the Virum Human project. So thank you, Professor. And we will open the session of question and answers. So please post concise questions in the chat, or you can open your microphone and ask Professor Curtis.
Okay, yeah. Thank you for such a great and inspiring presentation, Dr. Curtis. It really gave me new perspectives. So I have a question. Do you think metabolic outputs can be integrated with GWAS results to connect novel microbial proteins with host phenotypes such as COVID-19 severity?
And can you say the very first part again? What outputs integrated with GWAS?
You talked about meta-wibbly. I don't know how it's pronounced. I can type it in the chat.
Oh, Metavibula.
Yes.
The organization. Okay. It's a German word, and I'm sure I don't pronounce it correctly either. That's an interesting question. Certainly. Okay. Yes, I can think of a way to do that. There have been many studies that connect different quantitative measures from the microbiome with GWAS to help associate microbiome features with human genetic features. Usually those focus on taxonomic features, so which bugs are present, how abundant they are, and so on and so on. Even with that, where there's order of magnitude 1,000 taxa in most gut populations, even with that, your power can be very challenging because you're associating 1,000 different microbes with millions and millions of potentially millions and millions of different SNPs. So there are different methods that will help reduce that dimensionality and achieve good enough power in reasonably sized populations. If we wanted to do that for microbial gene function, so say microbial proteins or SNPs that are associated with human proteins or SNPs, then the dimensionality is huge. There have been papers that publish that, and I'm always real curious because the population size you would need to do that reliably is hundreds of thousands of individuals. Some studies which are just now starting to get there. One of the ways you can improve that and achieve good enough power in smaller populations is by prioritizing which proteins you care about on the microbial side. So we have not done it, but that's a way that you could put those two things together. So if you had a GWAS for COVID response, for example, you can use that to reduce the number of human proteins and SNPs that you care about. You can use something like MetaVibola to reduce the number of microbial proteins or genetic regions that you care about. And then you'll have a much more well-powered collection of features to associate with each other.
Thank you so much.
I
So,
hope that actually
it
answered the
happens
question. Yeah.
to Yeah, it happens to be actually my thesis work. I worked on
Great.
microbiome of COVID-19 and then did a GWAS of COVID-19 patients too. So
Okay.
maybe I can do this with my
I
data.
hope my speculation is similar to what you're actually doing. We did not do anything like that. So.
Yeah. Thank you.
Thank
Great.
you. Thank you, Sophia. Thank you. Thank you, Professor. So, your update on the human virome and how viral communities interacts with hosts and bacteria was especially insightful. So this is new. So I would like to ask a question. It's more about method. So what is the minimum workflow you trust to profile the virome?
That's also a really interesting question. We've been having conversations like that a lot inside of the HVP group recently. It depends a lot on what you want to do, since there are already lots of different ways that biological questions that one can be interested in for bacterial communities. There are probably even more that one can be interested in for viral communities. So one of the big differences that we see, the assembly-based methods that have been developed up till now are really good for viral discovery. So if you have a collection of samples and you want to really just find as many new viruses as possible there, you can VLP enrich them, throw out everything that's not a virus, assemble those, and most of the deep learning based classifiers for viral mags are really good now, especially if you do that with long read sequencing. You can find lots of very high quality novel viral genomes that way. But the sensitivity is not great. You won't find all the viruses in any given sample. You'll just find lots of viruses from the whole population. And you won't be able to quantify them. You've thrown off your quantification, so you know the virus is there, but you don't know what its copy number is, essentially. The problem that we're solving with baklava is sort of the opposite. We can never, by definition, find really novel viruses that way, but you don't have to do any special prep for your samples. And we're reasonably confident that we're evenly quantifying all of the viruses that we do detect across samples. And in well enough studied environments like the human microbiome, we're also reasonably confident we find most of the viruses because we're standing on the shoulders of giants and using a lot of these assembly resources that have been built in the past. So it really depends what you want to do. The two workflows are quite different.
Okay. Thank you, Professor. Because I'm working on the relation between the gut microbiome and colorectal cancer. So we do only the bacterial taxonomy. So it will be good to go further, like doing the archaea, the microbiome, and the virome also.
I didn't show data from it today, but we're just looking at viral profiles in Nicola's collection of colorectal cancer metagenomes to see what we find there.
Yeah. Thank you, Professor. is there any participant would like to ask a question to Professor Curtis? So this is a great opportunity to talk with Professor Curtis.
I have to do some mental math to figure out what time it is there. At least it's not the middle of the night. So that's
It's 3 p.m.
that's not so bad. I see one question in chat, it looks like, and thanks so much, Zakia. Again, I'm sure I won't pronounce names right either. Apologies. Regarding the Human Virome program, how will clinical metadata be integrated with virome data? And how will the program distinguish viral passengers, that viruses that have a functional impact on health? So the first half of the question is easier than the second half. For clinical metadata, that's something that we, and especially Owen, have been working on a lot at the coordinating center. Owen and his team have a lot of experience with building storage infrastructure and web portals for structured data. So they're just now finishing putting together a controlled language metadata schema for the metadata that's coming in from the different VCCs, the data generation sites. So we'll have a more or less unified schema for the different metadata collected among their different populations. Those will be shared both downloadable and through a web portal that Owen's team runs. He's not on the slide, but we're also working with Nikos Kirpides and the JGI team so that they'll have a special customized version of IMG specifically for the HVP data. So the first half of the question, we have a plan. The second half of the question is very difficult. I'd say in the absence of interventions, no computational methods work really well for distinguishing drivers from passengers in microbial communities because the arrow of causality can always flow in either direction. Genetics is a sort of privileged case where you only get causality in one direction and not the other. So channeling Wendy to some degree, a lot of what her lab does is either humanize gnotobiotic mice. So take microbial communities from people, put them into mice, and see if that causally transfers a phenotype, or even in even more detail, put a controlled microbial community into mice. So say a synthetic community, perturb that community with a specific added microbe or knocked out gene or other very controlled perturbation, and see if that then induces a phenotype. So I would argue that for like really showing causality, you need an intervention experiment of some sort, whether it's in mice like Wendy does, or in people if you do a controlled feeding trial or a clinical drug trial or something that introduces a controlled change.
Thank you. Thank you, Professor Huttenhower. Thank you for your time and thank you for your insights. And thank you to all participants for the great questions. Is there any more? Yeah, I think Zakia has more questions. Yeah, she has.
Time, I'm happy to, yeah.
Yeah, she has another question. So she's saying, are there ongoing collaborations with other global cohorts, like in Africa and in Asia, to capture virome diversity across populations? We're interested. We are very interested.
Yeah, of course. That's also a bit of a tough question because it's very heterogeneous. At the time of the original human microbiome project, there was a good bit of coordination between different national science infrastructures across the globe. There was and still is a big difference in available funding at the time. So some countries were able to fund really big initiatives, some countries weren't. There was a lot of willingness towards data sharing. So especially for the HMP1, many of those data sets were specifically designed to be globally available and circulated. The same thing is true for the Virome program here now, that the study is designed specifically to share data. but there's been very little coordination with international, like other national science organizations, for a variety of reasons. So we'll still be able to get the American data out. There's not as much official international participation. There are a lot of other more localized programs. So we've had a lot of luck with sort of East Asian cohorts, where sometimes on mainland China, but often in, Hong Kong, Singapore, Taiwan, the kinds of big cohort infrastructure that has been put together there is often able to integrate well with some of these programs. We've had interest from African organizations, as some of you may know. I was speaking with one of the continent-wide African microbiome programs a few weeks ago now. To my knowledge, there's not been funding available for a similar program in Africa. It would be great if there was, and it was arranged locally, similar to things like H3 Africa have done, where the funding is really put into doing the work and building the infrastructure locally so as to respect the populations where the samples and data are coming from. I think that would work great if there's a way to build more of that infrastructure and get more of that funding involved. Well, Wellcome Trust has been fantastic in doing that for some of the African genetics programs, for example.
Yeah, we hope so. We hope that we will have in the future a program to collect our microbiome, our proper microbiome.
We're having other problems with science funding in the US right now too. So I wish there was more that the NIH could do to help, but they're struggling right now.
Yeah, maybe in the future. Yeah,
We'll get
it's a
there.
perspective. Yeah. Yeah, before we close, is there anyone have question for Professor Huttenhower? I think that's it. So before we close, I wanted to say, please save a date. That will be in October. So we will host a microbiome day at University Mohammed VI of Sciences and Health. And it will be focused on neonatal microbiomes and also colorectal cancer microbiome. So it will be a full day of talks and sharing results and project showcases. And we would be honored, Professor Curtis, to invite you again to share your perspectives. Yeah, and to bring fresh angles and practical frameworks that move the field forward. So for the participants, we welcome talks, posters, and or simply come to learn and connect. So the official announcement and the registration will be published very, very soon. And if you would like to be notified to participate, please leave your name and the email in the chat box. And thank you all for joining. Thank you very, very much, Professor Curtis. We are really honored to have you today, Professor. Thank you very
Likewise,
much.
thank you. I'm looking forward to the next one.
Please, before going, can we take a snapshot of...
Yeah.
Yeah. If anyone would like to turn on their cameras before.
We had this problem all during COVID teaching. I would say, hey, let's take a screenshot. No one wants to turn on your cameras. So
Yeah.
it's okay to turn on
Yeah.
your camera.
So to who would like to turn the camera, we will take in a screenshot.
Okay,
Soumaya,
we
you'll
can see
take
you.
the screenshots.
Yeah. Say cheese. Thank you. Thank you very much. Thank you all. Thank you, Professor
Okay.
Curtis, again. And
Likewise.
see you next session.
Fantastic.
Thank you, Professor.
Bye, everyone.